Good afternoon, everyone. Another zebrafish talk. I'd first like to thank Francesca and Simona for inviting me over to Rome. It's very exciting to be at this excellent conference. And I'm going to tell you about some of the work that's been going on in my lab, where we've been developing zebrafish models to study COL4A1-related disease. So the main research focus of my lab really is intracerebral hemorrhage, or ICH, and I don't need to spend too much time on this for this audience, but we know ICH is the second most common type of stroke. But although it's not the most common type of stroke, it is the most deadly, so it accounts for around 6% of all global deaths. There are a number of risk factors associated with ICH. Probably clinically the most important is hypertension, but of course there are all sorts of underlying brain malformations which increase the chances of ICH, such as cerebral arteriovenous malformations, cerebral amyloid angiopathy, and of course, cerebral small vessel disease, including the monogenic types, such as COL4A1. Unfortunately, we have no specific therapeutics for ICH patients, so there's an urgent requirement to increase the translational research capacity if we're ever going to be able to improve therapeutic avenues. So one of the several or many reasons we have no specific therapeutics for patients has been a historical breakdown in the kind of preclinical drug development pipeline for ICH. So historically, we've relied heavily on rodent models for ICH, and most of the commonly used models for ICH are requiring invasive surgical procedures to induce the bleed in the brain. So although we're fairly successful in treating ICH in rodent models, none of these are translating to the clinic. So it's well recognized in the field that we need to revamp and improve the preclinical pipeline for ICH research. So we need to do this by improving the existing models, but also developing new models to incorporate into a new revamped pipeline. So the hope is we can use these new model systems in a bidirectional approach, back translate from the patients as well, to continuously improve this system, to increase the reliability of future preclinical studies. And in my lab, in particular, has been focused on trying to establish zebrafish disease model in this pipeline. So I won't spend too long on this because it's been set up beautifully by the previous presenter. But there are lots of advantages of using zebrafish to study human disease. Of course, they're vertebrates, so they share significant genetic and cellular conservation with mammals. The embryos and larvae, which is what we use primarily for our research, are small. They develop very quickly, and they're produced in large numbers, so a really powerful way for getting large sample sizes. Probably the most important advantage is the fact that the embryos and larvae are optically transparent. So it means we can use simple microscopy to observe internal structures such as the brain. But also in the zebrafish research community, there's a very large range of transgenic reporter lines which express different colored fluorescent proteins in different cells. So if you couple this with the transparency, it amounts to a really powerful approach for studying cell biology in the context of a disease using live imaging. And also in the context of ICH research, there are particular advantages of using fish. So as I said, the commonly used rodent models require surgery to induce a bleed. But we have zebrafish larval models which exhibit spontaneous brain hemorrhage. So we have chemical models where we introduce a chemical into the water which they absorb, and we have genetic models. And these models display spontaneous brain hemorrhages within the first 2 to 3 days of life. So it's a really quick model. So this image on the left here is an example of one of our models. It's a double transgenic. So it has a green fluorescent protein in blood vessels and a red fluorescent protein in blood cells. And we can see in the middle of the brain here, blood that's leaked out of the blood vessels. So it's a hematoma in the middle of the brain. So we can use live imaging to watch how cells respond to this bleed. So this is a very simplified overview of our model. So We collect embryos at day 0, fertilized embryos. They very quickly develop, and they have a functioning cardiovascular system by around 24 to 26 hours post-fertilization. On day 2, we start seeing these spontaneous brain hemorrhages. So in the next 2 or 3 days, we can study these fish to see how this brain injury and this hemorrhage progresses, and also how it resolves. So a few years ago, we tried to establish ourselves in the stroke research community, and we wanted to try and convince the stroke research community that we can use zebrafish to study stroke. So we wanted to show that the pathological consequences of blood in the brain in a baby fish resembled what we see in mammalian models, but also in patients. So the first thing we looked at was brain cell death. So this is a reporter line for apoptotic cells. So we see fish that have a brain hemorrhage, which you can see here. When we switch to the fluorescent microscope, we see this characteristic cluster of dying cells, which we can quantify. And we see a significant increase in apoptotic cells in the hemorrhaged fish. This is associated with a physical defect, so we can track swimming. So these red lines are the tracks of a fish, individual fish swimming in a 10-minute period. And these hemorrhaged fish swim less than their non-hemorrhaged siblings. And we can also measure neuroinflammation. So we know inflammation is a key driver of brain injury after hemorrhage. So we have reporter lines for innate immune cells, and we see that there's an increased recruitment of innate immune cells to the brain after hemorrhage and increased activation. So taken together, these disease outcomes in zebrafish larvae after brain hemorrhage suggests that there might be a useful tool for studying this disease. So how are we using the model at the moment? So you can split my lab into 2, basically. Half of my lab are interested in the time after the bleed. So they're studying brain injury mechanisms, such as neuroinflammation. They're looking at how the hematoma resolves. And we're also doing some drug studies to try and identify candidate compounds which can reduce brain injury. And the other half of my lab is interested in what happens before the bleed. So in the context of risk factors. So we're looking at environmental risk factors, such as infection, but also genetic risk factors, and this is where COL4A1 comes in. Again, I don't need to tell this audience this message, that COL4A1 variants are an important cause of small vessel disease and ICH. Many of you in the audience have contributed to these papers, but there's a whole plethora of evidence now which show that COL4A1 variants are a really important cause of small vessel disease and brain hemorrhage. So this led us to a question of whether we could model cerebral small vessel disease and ICH in zebrafish via loss of COL4A1. So all the work that I'm going to show you from now on is work that's been done by Daisy Flatman. She's a PhD student in the lab, very talented. She's currently writing up her thesis. She was funded by the British Heart Foundation. So we used the CRISPR-Cas9 gene editing approach to try and generate loss-of-function alleles in zebrafish. It was a kind of loss-of-function deleterious alleles was a quicker approach in the first instance. So there are 2 key approaches you can use. So we inject a cocktail, a CRISPR-Cas9 cocktail, which contains guide RNAs and the Cas9 enzyme. We can inject this into fertilized embryos, and then we can grow up those injected fish to adulthood, cross them with wild types to identify germline transmitters, and then once we've identified a founder, we can then create a breeding strategy and generate a mutant colony. The other approach, which is the quicker one in this instance, is to phenotype the fish that were injected, or the embryos that were injected with the CRISPR-Cas9 cocktail. So these F0 fish will be mosaic, so each individual fish will have lots of different mutations, but we hope the net effect will be a loss of function. So these F0 mosaic fish are nicknamed crispants. So the first part of Daisy's project was to characterize the COL4A1 crispant model. So the first thing Daisy wanted to do was to confirm that we were actually getting a loss of COL4A1. So we looked at the transcriptional level. So using qPCR, we showed that the crispant model had a significant reduction in transcript. compared to controls. And we've also used a different technique called in situ hybridization, where we've used a COL4A1 specific antisense oligonucleotide probe, which we incubate with the fish, and then we stain for the probe, which gives this blue signal. So this blue signal we see in the top two panels refers to where COL4A1 is expressed, so in the brain and in some of the vessels. And in our COL4A1 crispr model, we see a dramatic reduction in this blue signal. So this, taken together, suggests that we are getting a loss of COL4A1 transcript in this model. We were next, obviously, very interested to see if there was any evidence of brain hemorrhages. So when we looked at our live fish, so these are images of live fish down a bright field microscope, we were able to see in a proportion of the fish these bleeds. So you can see these reddy brown patches with the arrows. When we quantify this, we saw in the live fish around 20% incidence rate. We also did the same experiment, but using our double transgenic reporter line. So here we have blood vessels labeled in yellow and blood cells labeled in pink, and the bottom is a merge of the two. And we see that very clearly in a proportion of our fish that we have blood leaking out of the vessels in our ICH positive crispant model, but also in the ICH negative crispants, we see defects in the vessels and also reduction in blood flow. We did a qPCR on these crispants comparing hemorrhage positive and hemorrhage negative. And interestingly, the fish that had the most dramatic loss of COL4A1 expression were the ones that hemorrhaged. So we wanted to look at this cerebrovascular phenotype in a bit more detail. So we switched to a different reporter line called KDRL-EGFP, which gives us better resolution of the vessels in the brain. And we can see this very well-established, beautiful network of blood vessels in the brain of our control animals. But when we look in our COL4A1 crispants, we've got this sort of spectrum of cerebrovascular defects, some quite mild, some quite severe. So we saw in some of the milder ones, there was the odd blood vessel that was missing. There was enlarged spaces in between vessels in the brain. In the more severe cases, there was complete loss of some vessels and huge spaces. So taken together, these data suggest that loss of COL4A1 in zebrafish induces a spectrum of cerebrovascular defects. Given the role of COL4A1 in basement membrane integrity, we were next interested to look at, try to look at the basement membrane in our fish. So this is electron microscopy of a crispant, COL4A1 crispant brain. So trying to orientate you, this red line denotes a red blood cell within a vessel. This is the endothelial cell, and the blue line is what we believe is the basement membrane. And if you look closely at this basement membrane, we see these kind of breaches. So if you look at these in a little bit more detail, panels on the right, these kind of pouches of the basement membrane. We don't know exactly what these are. We still need to try and do some more experiments to understand them. But I think it confirms to some degree that the neurovascular basement membrane in our crispant model seems to be abnormal. So it's a kind of proof of principle experiment to prove that we were specifically targeting the COL4A1 gene with our CRISPR-Cas9. We wanted to do a rescue experiment. And to do this, we used full-length human wild-type COL4A1. So we cloned the full-length COL4A1 gene and in vitro transcribed to make mRNA, and then we co-injected the Cas9 cocktail with the full-length COL4A1. And instead of looking at live embryos, we decided to fix and stain our fish. So these fish, again, which was introduced in the previous talk, have been stained with a dye called the O-deanisidine, which stains for hemoglobin. So we can see in our crispant these brown, black regions, these are hematomas. So when we co-inject, actually the first thing to say really is that when we stain our fish, we were able to notice and record a higher incidence of hemorrhage compared to when we looked at the live animals. Possibly something to do with getting rid of the pigment. But importantly, when we co-inject with wild-type mRNA, we get a significant reduction in the incidence of hemorrhage, suggesting that we are specifically targeting COL4A1 with our CRISPR. We then went on to do the kind of opposite experiment. So we used site-directed mutagenesis to create 2 different pathogenic variants in our cloned wild-type COL4A1. So we made these 2 variants, this frameshift and this missense variant, and we repeated the hematoma experiment with the staining. And again, we show that wild-type COL4A1 was able to rescue our crispant phenotype, but both pathogenic variants failed to do so. And we also did the same experiment, but looked at the vasculature. So again, our COL4A1 crispant model showed these aberrant vessels, these large gaps in the vascular network, and this could be rescued partially by our wild-type COL4A1, but not by our pathogenic variants. So because of the neurovascular phenotype, we sort of hypothesized that there was a defect in angiogenesis in this model. So we devised a panel of qPCR probes, which were designed to target various angiogenesis-related genes. And when we did the qPCRs, and the only thing that stood out really to us was MMP9. So we saw increased expression of MMP9. So this is matrix metalloproteinase 9, involved with matrix integrity and regulation, but also things like inflammation. So we confirmed this in another experiment. So although variable, we do see a significant increase in MMP9 expression in the crispant model. We then did a pharmacological experiment where we used the MMP9 inhibitor, MMP9 inhibitor 1, and we treated our crispants. And the other group was treated with a vehicle. And then we stained the fish and measured the ICH area. And when we did that, we see that the MMP9 inhibitor was capable of significantly reducing the hematoma area. So taken together, these data imply that MMP9 may be involved somehow with the neurovascular phenotype seen in this model. So to summarize the crispant component, so mosaic loss of COL4A1 in zebrafish induces brain hemorrhage and neurovascular phenotypes, which are reminiscent of the human disease. Our genetic rescue experiments validate gene targeting specificity. Inhibition of MMP9 seems to reduce ICH severity, which might suggest targeting MMP9 could represent a candidate therapeutic avenue. But of course, there are some disadvantages of using the crispants. So they are mosaic animals, so there will be differences, genetic differences between individual fish, so that introduces variability into your assays. But also, we have to physically inject the embryos every time we want to do an experiment, so there's more manual labor. So Daisy next embarked upon a breeding program to see if she could develop a stable mutant line. So again, going back to this figure, she raised her crispants to adulthood, found a founder with a mutation of interest, and developed a breeding strategy to generate a colony of mutant fish. And after a while, Daisy found a really interesting mutation. So she found a 20 base pair deletion in exon 8 of the zebrafish COL4A1 gene. After heterozygous incrosses, she saw that the genotypes were produced in Mendelian ratios, shown by this PCR gel here. We made the mutant line on our vessel reporter background, so we could look at the vessels, obviously. And what we noticed, there was some subtle neurovascular defects in the model. So there was a slight increase in this gap in the network, the vascular network. There was thickening of some of the vessels as well. But overall, a much milder phenotype than the crispant. And of course, we're interested in looking at the hemorrhage rates. So we noticed that following a heterozygous incross, we were only seeing around 2–3% of our homozygotes that had spontaneous brain hemorrhages. So we next wanted to determine whether they were more susceptible to brain hemorrhage. So we used a chemical approach. So we used a drug called atorvastatin, which is a well-established drug in the zebrafish brain hemorrhage community for inducing brain bleeds. So at the higher doses of atorvastatin, all fish will bleed, specifically in the brain. But at lower doses, wild-type fish shouldn't really hemorrhage very much. So we did a heterozygous in-cross, and we were blind to genotype, and treated them with a low dose of atorvastatin, and then fixed them, looked at hemorrhages, and genotyped them. And what we saw is this massive skew in the Mendelian ratio. So over 50% of these embryos were homozygous mutant, suggesting that the mutants are vulnerable to brain hemorrhage if pushed with an environmental stimulus. Although the neurovascular phenotypes were more subtle and milder than the crispants, we did still notice there was a swimming defect. So we tracked swimming behavior over a 10-minute period and noted that the homozygotes swam significantly less and significantly slower than their wild-type siblings. And again, we wanted to look at MMP9, see if this phenocopied the crispant model. And indeed, we see a significant increase in MMP9 expression in both hets and homs. And now, towards the end of her PhD, Daisy has started looking at the at older fish, so fish in adulthood. So these images here are fish at day 100. They appear to be relatively normal. They behave normal. They even breed. So we had heterozygous in-crosses, and Daisy kept genotyping them and kept getting Mendelian ratios, so they're viable, they survive. But these fish did have shorter, so the homozygous mutants were shorter than their wild-type and heterozygous siblings. But also, interestingly, they had smaller eyes. We're not eye experts, we don't work on the eye, but I guess this might be interesting to some of you. And then most recently, we've observed spontaneous brain hemorrhages occurring in our adult fish. So just through sort of day-to-day observations in the aquarium, Daisy started noticing that a few of these fish had these red patches on the head, which obviously suggested to us that they may be brain hemorrhages. So she genotyped them, and they were all mutant. So we dissected some of these brains. So this is the skull cap of a fish, and you can clearly see this hematoma. This is a subarachnoid hemorrhage. We also wanted to look in the brain tissue. So these are wild-type and heterozygotes. There was no obvious bleeding phenotype in these brains, but when we looked in the homozygotes, we saw the presence of these patches in the brain tissue. So large dilated blood vessels in the brain tissue, and also hematomas as well. So these hemorrhages were visible in free-swimming fish. So they hold the potential for us to be able to watch the fish and accurately define exactly the onset of hemorrhage. So to summarize the mutant work, the germline mutation of COL4A1 in the zebrafish phenocopies the crispant model. We've observed neurovascular deficits in both early and late life. And these adult onset brain hemorrhages, which are visible in free swimming fish, may provide a useful platform for translational COL4A1 research. And to conclude, so loss of COL4A1 in fish recapitulates key aspects of the human condition at different stages of life. The embryos and larvae are excellent tools for live imaging of disease biology, and also hold the potential for high throughput drug screening. And the adult fish represent an excellent tool for studying the neurovascular deficits in fully developed animals, in mature animals with mature blood-brain barrier. Some of our data potentially implicates MMP9 as a candidate therapeutic target, but we obviously need to do a lot more work to understand what this is. And my take-home message really is that zebrafish represent an important new disease model and should be added to the preclinical pipeline for translational COL4A1 research. I'd like to thank everyone in Manchester who has been involved with this study, the guys in the lab, but particularly Daisy, who's basically presented a whole PhD, the funders, and thanks for listening.
Thank you so much. Thank you so much. Is there any question? It was extensive. There's a question over there.
Very nice work, impressive PhD thesis, really. I have a question about the rescue experiment. So you have this mosaic fish that you, where you inject also the mRNA, and you also inject not just the wild type, but also the mutant. I was wondering whether you could do some biochemistry on the mutant protein expressed, just to see whether in the absence of the wild type protein, it behaves differently, or as described beautifully before, it gets stuck in the cell or something like that. Thank you.
Yeah, excellent suggestion. It's something we've thought about. We've been thinking about it specifically for Daisy's PhD defense. It's something we haven't done, but it's something we would like to do. So yeah, excellent suggestion. Cheers.
Thanks.